This
Good to
conference
see
will
everyone
now be
again.
recorded.
Um, so I'm Josh Katz. I'm a scientist at DuPont in the Nutrition and Biosciences business, the Pharma Solutions business within that. We as a business predominantly supply to the oral solid dosage market, so mostly small molecule, but we do have a small team. Both Sue is on the call today as well, and myself and a few others who are focused on developing new technologies to really help with formulation and processing in the biologics arena. This talk today is not going to focus exclusively on our work. I do have one slide that talks about the surfactant we've been developing, and I'm happy to talk more about that. But the goal here today really is to talk very broadly about the alternative surfactants, and I'm going to use the term surfactant very loosely today for this presentation. So really about different stabilizers for biologics. So with that, and I will say, please do feel free to interrupt me throughout the presentation. The point is for this to be a discussion, and so happy to take questions as we go through this, hear comments, etc. Of course, we can then have more time at the end for that as well. So I want to start where we, by picking up where we left off at the meeting a week ago, where we had a very nice discussion about mechanisms of protein aggregation. So I believe that this very figure here was used in the presentation last week. Maybe it was a similar one from a different publication by Chris Roberts. But it's very important as we think about how excipients stabilize a biologic to understand what the fail points are and what the pathway is for that biologic as it goes down from its active monomer state to whatever type of aggregate you ultimately end up with, be it a soluble aggregate, a subvisible aggregate, or visible particles. Whatever it may be, what pathway it goes along is incredibly important in terms of your choice of an excipient and how these different alternative excipients perform in terms of stabilizing the active. When we think about surfactants, generally speaking, there are two mechanisms that surfactants are understood to stabilize biologics. One is one of competitive adsorption, where surfactants, by their very nature and design, are surface active molecules. And the idea is that they will competitively adsorb onto a surface. Oftentimes it's the air-water interface or an air-vial interface, water vial interface, a water IV bag interface. And the surfactant will competitively adsorb to that surface, thereby blocking the protein from accessing that surface, where the protein might undergo some part of its aggregation pathway. The other mechanism is one of preferential association. This is one where you actually have a direct interaction between the surfactant and the protein or a portion of the protein to provide some type of steric stabilization to the protein. Perhaps there's a hydrophobic patch that needs to be stabilized in solution. And so you have a direct interaction between the protein and the surfactant. Generally speaking, competitive adsorption classically is thought to be the dominant mechanism by which surfactants stabilize proteins, but certainly both are at play. And as we'll see as we go through this talk today, different excipients that are used might have more of a competitive adsorption or might have more of a preferential association mechanism. So this is, again, where understanding the path and the fail points of your protein is very important for, therefore, understanding which excipients might be better for the protein in question that you're looking to stabilize. So this is a talk about alternative surfactants. So let's start, though, by talking about why do we care about alternative surfactants to begin with. And so this slide here is all about the problems with polysorbate. Polysorbate, as we all know, is used quite a bit in formulations today. By and large, polysorbate 80 is what is used. Polysorbate 20 is used as well. Those two probably account for 95% of the formulations, at least, that are out on the market today with a surfactant. However, anyone's been reading the literature recently, anyone who's been to any of the major conferences recently is well aware of the fact that there are a lot of issues with polysorbates. So this slide focuses on polysorbate 80. Many of the same issues are true for polysorbate 20. One of the major ones, there's been a lot of literature recently, is the fact that the polysorbate has this ester bond here, which is subject to hydrolysis, especially as protein formulations are being driven to higher and higher concentration. There are higher levels of residual esterase and lipase that come through from the process in those formulations, which can chew up the polysorbate. Of course, once the polysorbate's been chewed up, it's not there to perform its function anymore, and the byproducts of the polysorbate, especially the fatty acid, can be a nucleation point for protein aggregation. Polysorbate is also a very vast mixture of different materials. So shown here is the USP monograph for polysorbate 80. This is just broken down by the different fatty acids that can comprise the polysorbate 80, but the monograph is generally silent in terms of where the hydrophobe, the fatty acid, can attach to the hydrophile, the polyethylene glycol sorbiton. So as a result, you can have substitution here or here or here or here, and you can also have di-substituted and tri-substituted products. You can have mixtures of fatty acids. So you have literally hundreds of different molecules in your polysorbate formulation. Polysorbates are also well known to oxidize. For polysorbate 80, it can happen through the unsaturation in the hydrophobe. You also can have oxidation of the polyether, the polyethylene glycol, which is well known in the literature. And finally, with all these problems, you ultimately still end up with a formulation that produces particles. So the polysorbate 80 or polysorbate 20 simply do not always work well enough for stabilizing the protein. And therefore, we have many different reasons for needing improved, better surfactants for protein stabilization. So there is a challenge, however, for surfactant replacement, and that is the balance of regulatory risk with your performance differentiation from your polysorbate. So generally speaking, the more you differentiate from your polysorbate, and that could be differentiation for better or worse, depending on the protein and how it interacts with the excipient, but the more you differentiate from the polysorbate, generally speaking, the regulatory risk involved is going to go up. The ideal case is that you have very good differentiation from polysorbate, some type of magic bullet that works great, but carries no new regulatory risk. Unfortunately, that ideal does not exist. So what I'm going to do today is I'm going to basically walk us along this arrow here, starting by looking at things that carry relatively low regulatory risk, but also do not differentiate themselves particularly well from the classic polysorbates. And we're going to move along this path, looking at things that differentiate a bit more, that carry a bit more regulatory risk, all the way down to the truly novel excipients, where we will see some very nice differentiation, but we also have to recognize that there is regulatory risk involved. And so for any protein of interest that needs formulating, you really have to look at how much differentiation you need, and therefore how much regulatory risk you're willing to accept as part of that formulation. So let's start down here in this corner with higher purity polysorbates. So relatively recently, we've started to see higher purity polysorbates being produced by a variety of different producers. Some example data here. So the image in the top right corner is from the product bulletin put out by the NOF Corporation, where they market a polysorbate 80. And one of the things that they highlight is their polysorbate 80 is completely clear, whereas a conventional polysorbate 80 is yellow in color. So there are a few different companies that are marketing this. Some of the benefits, they have much tighter specifications and less batch-to-batch variability. These are polysorbates that are designed to conform to the Chinese pharmacopoeia, which requires greater than 98% oleic acid content in them. And because there are fewer trace metals and other residual impurities in there, generally speaking, these polysorbates have higher oxidative stability than the conventional polysorbates. However, at the end of the day, the performance of these polysorbates is still similar to standard compendial grades. So I give an example here. This is a paper that came out earlier this year, looking at the regular polysorbate versus the Chinese pharmacopoeia grade polysorbate. And basically what it found is there really is no differentiation in the performance between those two grades. So if you have a problem before with your polysorbate that's unrelated to the impurities in the polysorbate itself, you probably still will have that problem. So if you have residual esterases in your sample that's chewing up your polysorbate, you're still going to have your polysorbate getting chewed up. If the polysorbate in general is not good enough to stabilize your protein, it's still not going to be good enough to stabilize your protein. That said, if your issue is just you were bringing in too much trace metal or you had too much batch-to-batch variability, this could be a solution for you. So let's now step to some other compendial excipients that are out there today. And so I want to start with Poloxamer 188, and I really struggled whether to call this a novel excipient or not, because at the end of the day, while it is not used broadly within our field, it is used today in our field as well as in other areas of pharmaceutical formulations. So that's one of the key advantages of Poloxamer 188. It's used today in injectable biologic products. It also is used quite a bit earlier in process for cell stabilization. So the upstream process for making the biologic, Poloxamer 188 is often used. It's often then washed out of the formulation as part of purification. This is a polymer that has no ester bond, and therefore you don't have to worry about ester hydrolysis. And it's able to generally directly stabilize unstable regions of proteins. However, Poloxamer is a much weaker surfactant than polysorbate, and therefore is a very poor interfacial stabilizer. So if interfacial instability is the problem that you have, Poloxamer is unlikely to solve that problem. You also see a lot of examples where Poloxamer is used at much higher use levels than polysorbate is, sometimes an order of magnitude higher. You do also have ether bonds still present. The chemistry there is the same as what you see in the polysorbates, so you do have potential for oxidation, though there's a lot less literature out there about oxidation of the Poloxamer in biologic formulations. And also, if you look at the formulations out there, it tends predominantly to be used in non-antibody biologic formulations, which I thought was interesting. So I do give you a few different select marketed products that I was able to find. These tend to be enzymes. Orencia is a fusion protein. You see human growth hormone on there as well. You can see some of the use levels ranging as high as 8 mg per mil for the Orencia, which is quite a bit higher than the typical use levels that we see with the polysorbates, which are in or around 0.1 mg per mil. So back to the mechanism, the polysorbates are generally understood to preferentially adsorb to the water hydrophobic interfaces. The Poloxamer is generally more understood to be a preferential association pathway where the Poloxamer is interacting directly with the protein. And this really goes to show how understanding the mechanism and pathway your protein takes could impact whether or not you're better off with a polysorbate or a Poloxamer for your formulation. The next example I want to give is the beta-cyclodextrins. This recently has been launched under the Kleptos trade name by Roquette. It's a hydroxypropyl beta-cyclodextrin that they are marketing specifically for biologic stabilization. Cyclodextrins have already been used for decades in parenteral products, typically they're used as solubility enhancers. The mechanism by which they work classically is that the hydrophobic cavity can work to help solubilize water-insoluble species into an aqueous environment through an inclusion complex. More recently, it's being explored for biologics, and Roquette has put out quite a bit of literature on this, including several different case studies. So one, for example, is that they've shown that they can reduce aggregation using 100 millimolar beta-cyclodextrin. But it was interesting to note that they also actually reduced the baseline amount of aggregate at the T0 time point in an agitation study. So if you look at the data here, you see with the 100 millimolar cyclodextrin, either the HP or the HPB, in both cases, after 24 and 96 hours of shaking, you see a lot less soluble aggregate present. But at T0, you also see less soluble aggregate present, which suggests that there might be some type of mechanism going on where the beta-cyclodextrin is interacting very directly with the protein to form some type of complex. While this is a talk about alternative surfactants, the beta-cyclodextrin has very minimal surfactancy. They gave a talk on this work at the AAPS meeting last fall. And the topic of surfactancy came up, and beta-cyclodextrin essentially is not a surfactant, though they're looking at it and using it in ways similar to how we use surfactants today. As a result, the mechanism of stabilization is probably very different, but therefore, again, understanding your mechanism, there might be utility for the beta-cyclodextrin. The next example I want to give is hydroxypropyl methylcellulose. This is a DuPont product under the trade name METHOCEL. I will caveat this slide by stating that the data I'm about to show you was not produced by DuPont. We were not involved in this. It was simply our product that was used and bought out on the open market. So there was a patent granted last year to reform Biologics, where it claims a formulation comprising an antibody and a hydrophobically modified cellulose polymer, specifically looking at our METHOCEL products. And what they found was that low viscosity grades of METHOCEL were required, and that our E-chemistry grade, that has to do with the hydroxypropyl and methyl substitution on the polymer, was found to be the best. So they showed some examples in the patent. For example, they had reduced subvisible particle formation for abatacept and cetuximab after agitation. However, they did note that the METHOCEL did lead to some increased foam formulation. And here's an example where the METHOCEL was useful, but it didn't really differentiate itself from the Polysorbate 80. So if we look at the data here, the number of particles, we see 893 for the E3LV grade METHOCEL. The PS80 had 380 particles. So not that much differentiation, or really no differentiation between the two. I should also point out that while METHOCEL products are used extensively in pharmaceutical formulations, as I mentioned at the beginning, our business focuses mostly on oral solid dosage forms. We are not aware of any injectable products that contain METHOCEL today. So there would be potentially some regulatory hurdles there if you wanted to use a polymer like a METHOCEL in an injectable product.
Hey, Joss, can I ask a question?
Absolutely.
So what's the molecular weight of METHOCEL? Because the cellulose is not a degradable polymer. So for parenteral applications, the molecular weight would become important.
Yeah, so the grades used here are fairly low molecular weight. We do have some molecular weight grades that are low enough that they could, at least in theory, undergo renal clearance. We have other grades, that are millions of Daltons. That's not what was used here. These are grades that are, in the single thousands of Daltons. Okay.
Thank you.
No problem.
So maybe, Josh, one more clarification. If it causes more foaming, does that mean that it might be degrading proteins more readily because of a lot more air-water interference, or that's not known?
Can you say that? It didn't come come through so clearly at this end.
Let me try. Maybe my headset is not working well. What I was asking is, one of the challenges you are highlighting is increased foam formation.
Yes.
Does that mean that it is potentially creating a lot more air-water interface, and that might also mean that it might be more destructive to proteins?
Yes.
Is that known?
That is exactly the concern. And again, we don't have data to look at this because this is not data that we did. But the concern there is, yes, if you have more foam, you're creating more interface. And so depending on exactly how you formulate, if the METHOCEL provides some interfacial stability, but then you're at the same time creating more interfaces, what's that balance there? And it's certainly a concern that you might be creating more interface and therefore creating more opportunity for your protein to find that interface and start to begin its aggregation process there. So yes, that is the concern. So
sorry, I had one more question. This is Nidhi. I'm wondering if you are aware if there is any database for like toxicity and immunogenicity kind of examples of some of these alternative surfactants for biologics to help with the use to justify the use in trials.
Yeah, so for a lot of these materials, each company tends to keep their own drug master file on the excipients that they sell.
Mm-hmm.
So, we have a large drug master file on the METHOCEL products. I don't know what, if anything, is in there about parenteral use. We tend to keep those files available only under CDAs.
Okay.
And my understanding is other companies are similar in how they do that, but obviously provide it to the formulators as necessary if their products are going to be used.
Okay. So there's no company independent kind of databases.
Not that. So the one area where there is, and I have it on a slide in a few slides, is the National Institutes of Health, NIH, has something called ToxNet,
mm-hmm.
where you can put just about any material you want in there. And it's basically a huge database of tox results.
Mm-hmm.
And so I do call that out on one slide that I have coming up for one of the novel surfactants. The challenge there is it often aggregates data for a series or class of materials, right? So if we went into ToxNet to try to look for hydroxypropyl methylcellulose, the ToxNet might not differentiate between different molecular weight grades or different chemical substitution patterns that might affect the toxicity. So ToxNet could be a good initial screen point, but you would probably need to do some of your own studies as well if they haven't been done by the provider of the material.
Okay, thank you.
So the next example is polyethylene glycol or polyethylene oxide, or PEG. This is the most common hydrophile in the surfactant excipients, and it's also generally used extensively in pharmaceutical formulations. Pegylation chemistry is used today to extend the half-life of molecules and shield molecules from immune recognition. So there's some pegylated biologics on the market already. I'm not going to focus on the pegylated biologics, but rather using PEG itself as an excipient. So PEG actually is an incredibly weak surfactant. Its equilibrium surface tension is around 64 millinewtons per meter, but it has been explored as an alternative excipient. That work has come out of Genentech and Stanford. So I thought I would highlight it here. We would expect it to have similar oxidative breakdown as observed with the polysorbates, and generally does not appear to be effective at reducing monomer loss or particle generation. So the example here is looking at a decrease in monomer concentration from a how much decrease in monomer you have, and so lower is better in this case. You see PEG is quite a bit of decrease in monomer content, though PS20 also leads to a decrease in monomer content for the antibody that was studied here, whereas poloxamer did not. But when you look at particle concentration, the PEG, again, you had a lot of particles. The PS20 blocked particle formation, whereas the poloxamer was somewhere in between. Clearly, again, poloxamer and polysorbate, in this case 20, very different mechanisms. The poloxamer seems to block the monomer to dimer transition, but doesn't block the formation of subvisible particles. The PS20 doesn't block the monomer to dimer, but then keeps it from becoming subvisible particles. The PEG seems to do neither. A closely related cousin to PEG is polypropylene glycol, or propylene oxide. This is the same as polyethylene glycol, just as an additional methyl group coming off the backbone of the polymer. It is the hydrophobe in poloxamer, has also been explored as an excipient itself. Again, this is IP that has come from Reform, and they have IP granted on using polypropylene glycol in formulation with antibodies. They were able to stabilize cetuximab against agitation with molecular weights ranging from 425 to 2,000 Daltons. They found, again, the results were comparable to PS80, but better than METHOCEL and PEG. And they stabilized abatacept against agitation comparably to METHOCEL. PPG has very low foaming propensity, and another area they've really focused their IP, and a benefit that they call out is that PPG readily passes through UFDF membranes, which means you could potentially add it prior to your UFDF step and not worry about overly concentrating the polypropylene glycol or being able to easily remove it from your formulation. So I show here, actually, they use PEG as a non-working example in their patent, but the different polypropylene glycols here, you can see the DLS diameter does not go up with the polypropylene glycol, whereas it does with the PEG and a few other chemicals that they looked at. And again, the foaming, very low foam and a very fast dissipation time of the foam compared to the METHOCEL, which came up quite high and stayed around for a long time. So those are some of the excipients that are used today in the pharmaceutical industry, but maybe not for biologics. What I'd like to talk about next are some of the more novel excipients that have come out. I'm going to talk about 3 different classes of surfactants. I'm going to talk about the bridges, the alkyl maltosides, and the N-alkyl amino acid polyether amides, or FM1000, which is the focus of our group here in DuPont. So I'll start out with the bridge surfactants. The chemical structure of the bridge is in the upper right corner. It is basically a linear alkyl carbon that is then directly grown from that a polyethylene glycol through an ether linkage. So these alkyl polyethers, they are sold by Croda into various industrial and personal care applications. However, based on what I was able to find, aside from a few topical applications, they're generally not used today in the pharmaceutical industry. There's one oral tablet approved in Japan, but that's about it. So they aren't really used today, which is why I consider them to be, an alternative new excipient. The bridge surfactants contain no ester bonds, so they should be resistant to host cell proteins, though oxidation will, again, still be an issue, as is the case pretty much with everything we've talked about so far because of the polyethylene glycol. Several different companies have started exploring bridge surfactants as alternatives, and when they do, they typically choose surfactants that have hydrophobes and HLBs, hydrophilic lipophilic balance, similar to those of polysorbates. So in one example, Bridge 58, which is a C16 ethylene oxide 20 polymer, reduced monomer loss on agitation similar to polysorbate 80, but at lower concentration. So that's this data down here, where you can see that the data actually looks very similar. The black bars is the shake data, the white bars is just day 0. You can see the data is very similar to polysorbate 20 or polysorbate 80 for these 3 different antibodies. However, the level of bridge that is used is much lower than that of the polysorbate. So you can see 75, 25, or 12.5, whereas the polysorbate 20 is at 400 here, the polysorbate 80 is at 600, here it's at 400. Another paper also found that Brij 92 bound to recombinant HGH comparably to polysorbate 80, so they interact with proteins similarly. However, on the flip side, if you look at this paper that came out a few years ago in AAPS Pharma Tech, the Brij surfactant led to quite a few number of particles with an antibody in a agitation study compared to the polysorbates that are over here. Also, this is where I mentioned the NIH ToxNet. I did a search for Brij in the NIH ToxNet, and they had a reported LD50 of about 27 mg per kg in rat, which is lower than what you find for the polysorbates by about an order of magnitude. That could limit adoption by the pharmaceutical industry. I don't know. Again, 27 mg per kg is at the same time much higher than what typically would be dosed to a patient, but the fact that it is lower than polysorbates might raise some concerns. Next one is the alkyl glycosides. So these are, unlike most of the surfactants we've talked about so far, these are surfactants that contain sugar head groups as opposed to polyether head groups. And as a result, these do not undergo oxidative degradation like the polysorbates or the Brijs or the poloxamers. There's a suite of IP that was filed and granted to Aegis Therapeutics around the use of these surfactants. Their main focus is dodecyl maltoside, though their coverage is more broad than that. And they developed these surfactants for 3 different applications. One is as a mucosal permeation enhancer, specifically looking at nasal mucosa delivery. One is for protein stabilization, and one is for hydrogel formulations. Their lead application was the mucosal permeation enhancement. And after this excipient was approved with the approval of a drug called Valtoco, which is a diazepam nasal spray. It was the first time this excipient was in an approved formulation. Aegis was acquired by Neuralis, who makes that drug product. And it's unclear at this point if the Protect platform is still being pursued by Neuralis, but this surfactant is out there. There also was a filing by Genentech Roche on alkyl glucopyranosides back in 2011 that showed the improved stability of monoclonal antibodies to both agitation, heat, and oxidation. So here's one example from that where you see that with the surfactant you get very high monomer content over a long period of time, whereas without surfactant or with polysorbate you see a drop-off. Finally, talk briefly about the N-alkyl amino acid polyether amides, or FM1000, the structure which is shown here. This is a novel chemistry format that we and DuPont developed to address many of the limitations in protein stabilization. So to address the issue of ester hydrolysis, we built the surfactant with amide bonds. We would expect similar oxidation to the polysorbate, again, because we have a polyether, but we don't have the esterase concerns. Our molecule is chemically defined, so there's going to be low batch-to-batch variability, a little polydispersity with the polyether, but only a single hydrophobe is used. The FM1000 has rapid interfacial stabilization to outcompete proteins, and as a result, improved agitation stability across many proteins studied. It also can improve the thermal or solution stability of some proteins, potentially interacting directly with those proteins. So both mechanisms of aggregation, or both mechanisms of protein stabilization using a surfactant. And generally, you can use lower concentration compared to the polysorbates because of the fast interfacial stabilization that it provides. However, it is a completely new excipient. We are doing tox tests on this right now, so we are doing our own tox testing to answer your question from before, and we make that available under CDA, and it's not yet a commercial material. So there's just 3 examples on here. One is showing the rapid interfacial stabilization of the FM1000. It stabilizes the interface an order of magnitude faster. So if you're concerned about the protein getting onto an interface, this will kick it off that interface an order of magnitude sooner, giving it less time to go through whatever part of its aggregation pathway it might do on an interface. One example of improved stability is here with cetuximab. This is subvisible particles. The FM1000 has about an order of magnitude fewer subvisible particles than the polysorbate 80 after agitation. And if you look at monomer content following a heat age study of abatacept, you see the monomer content stays up about 97% for the FM1000, whereas poloxamer, which is the incumbent technology for abatacept or the no surfactant control, see about a 15% drop in monomer content.
Josh,
So,
how much of FM1000 is added in each of those examples?
Yeah, so for these examples here, this study here was done at 0.05 mg per mil surfactant. The data at 0.1 mg per mil is almost identical. It's very, very similar curves at double the concentration. This study here was done at 0.01 mg per mil surfactant, and this one was done at 0.1 mg per mil surfactant, I believe.
Thank you.
So to conclude, we have a limited but not insignificant number of novel excipients that are available and being explored for polysorbate replacement. This also, I should say, does not include anything that might be developed or evaluated by individual companies that they haven't published yet. So, for example, there was a talk at AAPS last year on an evaluation of a suite of like 30 different novel excipients, but those excipients were not disclosed. They might be ones that we've talked about today, they might not be. But, companies are working on that. Different excipients also have very different mechanisms of stabilization. So knowledge of the dominant mechanism or mechanisms of protein destabilization and aggregation is very necessary to help drive your choice of a replacement excipient. I also do want to highlight that the FDA right now is working on pathways for new excipients. As it sits today, an excipient can only be approved if it's used in a drug product that gets approved by the FDA as part of an NDA or BLA. However, they are working on new guidance to have a separate approval process specific for excipients. And hopefully that will lower the bar for bringing new excipients to market, which will enable us to better innovate to address the challenges facing our formulation community. So with that,
hey Josh.
Hopefully we can have some discussion. Yeah.
Hi, thank you for the presentation, but a quick question on your, not just the last comment, but also your previous slide, if you don't mind, the N-alkyl amino acid surfactants, this one here. So you said there are stock studies going on. This is within DuPont, you mean, or are there other companies doing this?
So DuPont is contracting for these studies to be done. So we aren't doing them in-house, we contract it out, but there are studies, we own the data, we're the ones designing the studies.
So you guys would be providing, as an example, you'll say, okay, this is a new surfactant, and these are the tox studies that we have actually done to show this is, workable solution for biologics.
Yes.
And then, of course, the FDA has requirements about a new excipient in the formulation, and they might actually do it the sponsor company would actually have to do those studies, right?
Right. So that's
okay.
Something that would depend really on the application of the FM1000 and defining, right, what studies need to be done, and how do those studies best fit into the clinical development in general of the formulation. And therefore, who should have responsibility for which types of studies.
Got it. Okay. Thank you very much.
Sure.
Okay, but we are, of course, open for any other questions for Josh. So please speak up.
So, hi, This is Vasco from Sanofi. Can you hear me?
Yes.
So I used to work in a company that was researching for novel excipients for reduction of viscosity, surfactant properties. And one of the so we spent several years investigating this. One of the issues that we had now talking about polysorbate replacement molecules. So one of the issues we found a lot of molecules that were better. If we would test different molecules and agitate it violently, there was a lot of them that were actually more protective. But one of the major concerns that we had, so one of them, if you had some binding, then of course it could destabilize the protein in conformational wise. And here I can see that you're showing some examples where this doesn't seem to be the problem, at least compared to some of the controls. But one of the major problems was also toxicity. So there were a lot of them that were good, but then when you tested in animals, then they were always more toxic than polysorbate. So I'm just wondering, how confident are you, and is there a new generation that actually ticks all the right boxes that you're confident that it will pass everything, all the tests, the regulatory tests that
Right.
are
No,
required?
it's a great point, and maybe that's I mentioned that with the Brij surfactants that might be a concern there. Without speaking to the details of our tox studies because they're still ongoing and those are proprietary right now. Suffice it to say, we are comfortable continuing to move on with the development of FM1000.
Mm-hmm. Okay.
So this does seem to check the boxes.
Okay, good to know.
So maybe to follow up, Vasco, on your point, and maybe a question to both of you is, as Josh highlighted, there are multiple properties we are looking at when we think about polysorbate or any other surfactant. So in terms of checking the box for any new surfactant, the list we are seeing here, is that pretty much covering all bases, Josh, as well as Vasco, from your past experience?
So I would say, from our perspective, when we look at FM1000, it brings a lot of the benefits that people need based on the challenges of the polysorbate. And at least at this point through our development, we are not seeing any red flags in our toxicology studies that would say, wait, we want to stop developing this. It's performing in a way and behaving in a way that we are comfortable with for developing it as an excipient. Maybe a different example, the other way to look at that is we go back to this slide here. What I didn't even highlight as a potential novel excipient, but that actually performed quite well in this study, in this paper here, is Trident X100. So Trident X100 is a surfactant that's been around for a long time. It's used for viral inactivation in bioprocessing, but it's also right now being regulated out, especially in Europe, even from the bioprocess. So the idea of putting that into a final formulation at this point, I think, would have such a high regulatory burden that I didn't even bother to put it into this presentation as a potential excipient replacement, though it might work quite well, even compared to polysorbates.
Right. Well, given that you brought up Triton X, the regulatory burden there is quite different from everything that you have talked about here, if I understand correctly, including concerns for environment and stuff like that. So that's not that's not one of the items you have listed here for any of the surfactants.
Sure.
But by the way, so another question I have on FM, yes, FM1. So this looks solid. Particularly, I like your number 3. That, as you described, is one of the major mechanisms that we believe polysorbate is helping us with, right?
Right.
That's the interfacial stabilization. So that one looks as if that's pretty strong. It's helping to prevent particle formation, and the concentration you're using is also pretty low. So yeah, it does look pretty solid to me. Question for Josh. We have another 8 minutes.
This is Danny. I have one question. My question is, what about the idea of not reinventing everything, but slight modification of the components of polysorbate? What it means, the fatty acid in the the surfactant group, instead of the ester bond, make it into say an imide bond, and therefore, like you said, could reduce the enzymatic hydrolysis rate. I understand that would still not eliminate oxidation risk, but oxidation, for the most part, can be greatly reduced by the proper storage and other methods that really can be controlled. Sorry, I missed the first part of the presentation. I was late. I don't know if you mentioned this.
So no, it's a really interesting question. It's one that I hadn't considered before. So basically, if you're suggesting, replacing this ester here with an amide instead.
Or yeah, something. Yeah, exactly. Something like that.
So I'm not familiar with the process for producing polysorbate, but my general understanding is the way that polysorbates are made is that first the sorbitan polyether is made. So you basically end up with a molecule that truncates here, at which point they then do an esterification reaction by adding the fatty acids in.
Mm-hmm.
So the challenge for doing that is finding a way to take one of these terminal OHs and converting it into an amine, and then doing at that point an amidation instead of an esterification. I'm sure chemists could come up with a route to do that. I don't know if anyone has thought to do that yet. And it's an interesting concept. I agree it would solve the problem of ester hydrolysis, and it should generally otherwise perform very similarly to polysorbate 80 without that issue.
Mm-hmm. Yeah, I just thought about, just a wild idea. Thank you.
Hey, Josh, Sridhar here again from Roshan and Tech. Very quickly on your new excipient, the FM1000, I think that's what you called it, right? The alkyl amino acids. Are there, what kind of amino acids are there? I see phenylalanine there. I don't know if that's what you meant by pH, but do you know what amino acids are there, or is it a different composition? Is it a mixture? Is it just purely based off of one amino acid?
Yeah, so we make it just with a phenylalanine.
Gotcha.
When we first did this work, this is the paper that we published back in 2016, we did look at a few different amino acids, and the one with the phenylalanine really differentiated itself. For reasons that are still not entirely clear to us, we're still trying to figure that out, but the FM1000 was much better. And so that's the one that we've moved forward with and continue to see good results with. So, as we have time, we try to go back and answer some of those why questions, but for the time being, we have this surfactant that works quite well. And so, yeah, we make it, it's a very straightforward two-step synthesis where we amidate the fatty acid with phenylalanine and then do a second amidation with this hydrophile.
Got it. And so are you guys coming to different companies for discussing this? What's your step forward?
Yeah, so we've published this, we've talked about this at a few different conferences, and yeah, we are now talking to different companies to present. There's a lot more on this than I'm presenting on the one slide, and we're happy to take conversations offline and talk with individual companies, put confidentiality agreements in place when appropriate, and see if and how FM1000 might be beneficial to each company's individual portfolio.
Got it. Yeah. Well, thank you.
No problem.
Josh, is CMC, critical micellar concentration, known for FM1000?
Yeah, so the CMC of FM1000 is about an order of magnitude lower than that of polysorbate.
So, for example,
so all the studies we're doing are well above CMC.
Okay, great. And one more thing is at, let's say, 0.01 or 0.05 mg per mil that you are adding here, the surface tension will be how much? Like 40? I can't tell from this graph.
The CMC, you can't get the CMC from this graph.
No, I mean it's a surface tension.
Yeah. Oh, the surface tension. Yeah. So the equilibrium surface tension for FM1000 is around 40 millinewtons per meter. It is going to vary on exactly what buffer you have, but 40 plus or minus 2, let's say, is your equilibrium surface tension.
Excellent. Okay, we still have 2 more minutes. Any other questions for Josh? Josh, maybe one more question, if you can entertain, as we talk about protein aggregation mechanism, right? And
yeah,
the very first slide you showed was how these surfactants are actually helping, right? So if you think about FM1000, is there any thought that is it working through your pathway A or completely avoiding pathway B, etc.?
That's a great question. So we actually have evidence that it does both. And that's one of the things that excites us about the FM1000 is that in some systems, it really outperforms polysorbate 80, where polysorbate 80 is otherwise the best surfactant to be using. And then we have other systems where it outperforms poloxamer, where poloxamer would otherwise be the best surfactant to use. So we do have experimental evidence that actually both mechanisms are at play with the FM1000, that depending on which one you need, the FM1000 is effective at both. This was surprising to us, but and it's something we want to explore further. Very much depends on each individual protein, right? Because each individual protein is going to have different pathways and therefore need different balances of these 2 mechanisms. But from what we've seen so far, the FM1000 seems to be able to participate in both.
Thank you. Okay, we are just about 30 seconds away. Any last call, any question? If not, Josh.
No.
Oh, go ahead, Sridhar. Do you have any questions?
No. Sorry, I was just saying. Thanks for organizing this. Thank you. That's all I wanted to say. Yeah.
Absolutely. And by the way, thanks to all of you for joining and asking really good questions and bringing your expertise to the discussion. And Josh, this is a really nice summary, sort of giving the overview of major surfactants that folks have been using. So with that, we close the meeting, and thanks to all of you again.
Thank you very much. Have a good afternoon, everyone.
Bye.
Thank you.
Thank you. Bye.
Bye.
Thank you.